Okay, many of you, the first time be in this room, and if you haven't been here before, you're supposed to be able to log in any of those computers using your NetID with at tamu.edu. Then let me know if you have a problem. We probably have to find the IT support.
I'm having issues.
Having issues.
Reset my password and see if that helps, but it's not working for me.
Okay.
It was same with that computer. I switched on the computer and it works.
Okay, so some computer may have a connection issues. Maybe you want to change it to another computer. Yeah, it will work. It happens sometimes. To get into this online syllabus, you just Google VIBS 675. This is a course number. This course number supposed to be updated in the system, but this year it still shows up as a 689. But next year we should use this official course number. So if you're correct, landing on this page, this is a updated page. So I keep everything update, correct room number, and here it's good. And on your new computer system right here in CVM, we start to use Microsoft Teams. And I create a team for this course particularly. And you should be able to click this link or this. The link is on the web page. The good thing about this is better than email because email get lost sometimes. But here you keep every message, especially once you request, I will approve, and you will become a member here. Inside, for each lecture, I will have updated links. I will put the PowerPoint slides if you need here. We may later use this for homework. I don't know how the system will work, but it could be. All right, so you can put a message as well if you like. So now I receive, for example, the request from Carissa and Victoria, and I will accept. You will be putting in here as a member. All right. We found this useful to keep information stay there and also updated. Occasionally, I have an announcement. I will try to send email to this email list as well at the same time. So this will be the email list for the course. Okay, you should get updated information. So that's this. It may take some time to let Teams to install if you use a new computer system here. Next, most important, we will take a look in Zen Network. It's here in this teal color button here. If you double click and search MATLAB, you should be able to see the icon. Anybody has this issue? So we need MATLAB as a main language in this course across the whole semester to illustrate and demonstrate everything. This is a huge system. It takes some time to install. What I'm going to do here, I will just double click and see there's a warning message. I will say OK to let it to install in the background. and it will take maybe 20 minutes at least. I just do nothing. I won't double click or just click once, then just wait. We do something else. Come back, it should be installed on your machine, okay? If you switch machine, because majority of this machine will install this, you should be okay next time. If someone else occupied your spot, it's fine. Mm-hmm.
I can't see the application inside the ZBrush. I can see the outside actually.
It's just not here. Not no MATLAB. You probably have already MATLAB installed. Yeah. So, yeah, you can use this older version. It's all fine because that's. But if you search MATLAB, it's here, so you can double click. Yeah. Yeah, double click. Your machine should have older version. It's 2021, but you can update. If you don't want to, you can leave it like that. The older version will still work. Yeah. Okay, so this is just some housekeeping stuff. Will take some time to let computer to set down. Computer take time to update itself. So many problem will solved by time. Okay. Any questions so far? We don't have a TA here, but I should be able to take care of all of you. Okay. Okay, I think I should give you a delayed welcome message and welcome to this course. So we will try to make biology students become more computational and using the language which data scientists or machine learning people will normally use to describe their algorithm, to understand the principle behind each step of the algorithm. And on the other hand, if you have a computer background, engineering background, I want you to become familiar with all the updated, the most exciting single-cell biology field, and you get into details and why people doing that, why biologists are thinking in that way. So once you have a data matrix, what you are going to do and how you can compare between samples, many questions. And for computer background people, you will start to think about the algorithms you know and how can you apply and translate, transfer your knowledge from the computer side and apply to biology questions. Okay, so this is like a double sides, and people, I hope you will enjoy it. And I will quickly go through our outline of this course. First, Tuesday was supposed to have the introduction of the global background, the principle, why you can get information out of individual cells is such small, entity, what's the technology you used for the barcoding system. Then we will very quickly go through those slides today, and I will have a quick review of the history of machine learning field, and that's the introduction. Then next week, we will start to look at vectors and matrix. So we discuss of the data points. Each data is defined by properties. Those properties, the values, create by a vector. So each vector defines a data point. And we will put them together and make a group of vector as a data cloud, okay, points. That's a matrix you can represent. So what we can do and what kind of vector matrix we have. Then in the same time, I will introduce how can you get from the data which using, for example, 10x genomics system to from raw data to become this data matrix, okay? That will be next week. Then we will discuss important, when you compare data points one against another, you will measure the similarity or difference between them. This is a fundamental operation you can do because a lot of downstream analysis depends on how you define the similarity or difference between these data points. At the same time, we will see how, on the biology side, what we can do once you have the matrix. Are you going to do the QC, and what kind of genes you want to get rid of, and what kind of cells you say, okay, it's a high quality cell that has been measured, and then you will keep in the downstream analysis. This week 4, we will see some of the data modeling, normalization method. There are a lot of problems with people are discussing what kind of normalization is better for what kind of condition. So it looks simple, but it's not that simple. In the same time, we will discuss probability and the distributions. Week 5, we start to do visualization because we are dealing with this high-dimensional data, and we will touch base on the most important topic, the PCA. Then you reduce the dimension and start to look at the relationship between data points. And once you reduce to 3 dimension, you will be able to visualize it, okay? There are many, many different ways you can do this. We will discuss the principle under PCA-Tisney UMAP fit, and you understand it's not just one click button and get the picture, but how exactly under the hood, what's going on, how the data points have been rearranged and put together. And then week 6, we are going to study clustering because this is the most popular or unsupervised algorithm like computer scientists really like. If you discuss clustering, they will discuss the whole day with you. There are many, many different ways to do the clustering, and we will use that, and we will go through several different clustering algorithms and which one is good, which one bad, why, and then apply to single cell data. Then we will start to do clustering and another very important topics in computer machine learning. We will also discuss the cell type identification once you have a class of cells, and then how can you define what cell type it is. And we will have a real data applications, marker genes identifications. Then week 8, we'll discuss specifically about the highly variable genes. That's a gene in the homogeneous population of cells. Once you know the cell type, then you get this particular cell type. So under your control, this group of data points are all from, for example, T cells. And then you see, okay, what we can learn from this single population group of cells without any other information, we will look at the variability of particular genes expression within this group of cells. Those highly variable genes are really important. It's a topic I really like to talk because this will against a lot of common sense from biology field. Biologists try to control the variability and try to get rid of it with increased population size, sample size, for example, to control the difference or variability across one sample against another. However, in single-cell world, the variability is the most important feature or characteristics that can be used to define the function of the genes to reflect the function of the cells. So we against the mean, we don't take average, we will look at variability instead. And then we will move to regression and regularization, and this is like related to feature selection. So you select which variable you should be used to explain another variable and how you select them. And we will have composition, decomposition, and SVD, ICA, and MF, non-negative factorization. We'll discuss bulk RNA-seq, single-cell RNA-seq. We will discuss pseudo-time analysis, and in the same time, another topic in computer science is optimization. How can you find the solution of a given equation or some of the landscape of the likelihood, and try to get a better solution once you have multiple variables, and how can you combine them. And then we will discuss the manifold alignment. There are several applications and use tensor as well. My lab is specifically working in this field. We have a series of tools have been developed using those algorithms. And we will then discuss how can you construct gene regulatory networks, another topics. Okay. Then this week, students will come to have your presentation and see, okay, from learning this, what you can do with your own data, or what you have a good idea, and combine some of this and put together, have a new idea about your project. We will have a lot of, through the course, I will have a lot of examples to tell you. For example, using clustering algorithm, plus some disease, non-disease samples, and how can you define which cell type will be the better clustering, and this cell type will be targeted for research. So there's a lot of combinations to put together to have a very significant biological question, and you can solve using the combination of those algorithms. It should be very creative. The final exam will be 30 multiple choice questions, and it's online, so you just click and submit. You don't have to be in the classroom. It's online, so I will send to you. And throughout the course, I will explain what kind of question I will ask. Okay, so mostly you have 4 options. One is correct, so it's really hard to construct those questions in reality. Any questions for this outline? No? Well, okay. So here, my MATLAB is still trying to install itself. So just wait. Yeah.
For the final project, do you require to have your own data, or the examples that you will be providing will be sufficient?
You can use existing data, other people published, but you have to re-engineer it and build a new story out of it. It's called a secondary analysis, but mostly we can see primary analysis has not sufficiently extract all the useful information from the data, but you can use your new algorithm, new idea, or you can combine 2 existing data sets together to answer some new questions. Okay, you will have some good ideas throughout the course. Okay. Mm-hmm.
It's asking me for admin access.
It shouldn't, because if you through the network, all the program here has been improved without using any administration privilege in order to install. Otherwise, you cannot install anything on those computers.
Okay. I'm not sure I understand the part.
Mm-hmm.
Sorry, I was dealing with logging stuff.
Yeah. Okay, let's see. This is a story in primary research institute in Tokyo University, I believe. And so this chimp has been trained once you look at these numbers, and the number once she touches one, and then the rest will be masked, that she will remember the sequence and get the food once you touch the sequence correctly from 1 to 10. So this is some ability. This is really hard to do. You try it. This looks like this chimpanzee developed a bit, have been trained using either supervised learning or not, and get some of this intelligence. And humans are different. They have a different ability in terms of intelligence. We tend to use tools. We across the human development, evolution, we start up as a Stone Age, and to use those tools for maybe million years or at least hundreds of thousands of years. The human tool use history into several ages. So this is Iron Age, and we are now in the Silicon Age, for example. Okay, this has greatly increased our productivity, and when say, okay, what is machine learning? Is computer science field. They give computer ability to learn without being explicitly programmed. Okay, that's an early definition of machine learning. One of the algorithms is called PageRanker, and this algorithm help these Google founders to build their kingdom right now. And this is an algorithm to evaluate the value of each node in this connected network. Okay, you have an input, output. You can iterate this and then find out the A is more valuable than other BCD. There's an algorithm to compute. It's pretty easy if you understand those. And then people start to ask whether PageRanker as algorithm is a machine learning algorithm. Because depends on the machine learning, you need to let computer to do something without explicitly program. So there's some argument whether PageRanker algorithm is a machine learning algorithm or not. There are some general definitions and classifications. We will discuss supervised and unsupervised learning and without touching reinforcement learning. This is most recently like Google Google DeepMind using a lot this to train the gamer. So unsupervised learning is data driving, for example, clustering without train the data, just light algorithm directly and do the clustering by itself. And other example like regression and classification is considered as a supervised learning because you have to start using some of your known data points to train the model, and once you finish your training and you can apply this model or you train the program to do to accept the new data points to classify it or regress. So this is like gingerbread graph, and you can find online easily, and also supervised, unsupervised reinforced learning. So this is an example of supervised learning. On your left is linear regression. You try to learn from these blue dots and found a curve, not a curve here, it's linear, just a line which goes through most of the data points, make sure the distance, the yellow, the deviation as small as possible. So we are fitting this line using slope and intercept in order to achieve the minimized this deviation, which this green, this yellow lines. You are doing is a training. So now if you have a new data points somewhere, for example here, then you will predict given X value and what the Y is supposed to be on average or by the train. So this is supervised learning. And support vector machines is a standard supervised learning. You use those classified data points, black and white, to find a separating plan to separate those data points. After you've found this line, the best solution in this case is this right line here to separate these two clusters. Then once you have a new data points come in, and you will classify it by either it's in the left or right side of this line. This is by the separation. So neural network is also a trained supervised learning machine, and you have to use some kind of training data in order to adjust or the parameters within this neural network to allow it to give the best output. And once you finish the training, you can start to use this to get the new input data and give you the results. And unsupervised algorithm, the PCA is unsupervised. You don't have to train data, just directly apply to your data points, to rotate it, and to let the data to show the most variable among the data points, variability. And the k-means algorithm is a clustering algorithm. It's unsupervised. Okay, a lot of applications in the daily world, and it's everywhere. There are some of the pipeline, and we just quickly go through. And this, the neural network is composed of the neurons, and the neurons is defined by this data structure. And you have an input, multiple input. Multiple input will be aggregated together, and then the neuron in the middle is a function from the aggregated information to calculate the outcome. So this is a very simple structure that is defined in 1943. And once you put a lot of neurons together, this become a trainable neural network. So this early development. So the neurons have been developed, the binary neurons, this data structure has been defined even before this this computer has been created. The Alan Turing started as this the intelligent machinery and the paper, and then Arthur Silmer, the one defined this general concept of the machine learning, developed this self-learning algorithm, the check program. 1963, support vector networks. So this is support vector machine, the early concept. Then Moore's law has been mentioned, and then 71, Stanford started to do this this vehicle, and then 79, convolutional neural networks have been proposed. Self-organized map, it's one of the unsupervised learning, and the Bayesian network, 82. Okay, simulated annealing, 83. This is a very important optimization algorithm, gradient descent, 83. This reproposed this support vector machine. It's been widely used in the classification field in the machine learning. Reinforced algorithm, and then 97, Deep Blue, and the chess beat this world chess champion. And 98, PageRanker algorithm proposed. GPU start to be used to implement the neural network. That's 2005. And this self-driving cars, and Li Feifei, and start to use this ImageNet database to train neural networks. Large scale and visual recognition, and the ImageNet. And 2016, and the AlphaGo has beat Li Sedol, which is the Go master or the champion. This is just very brief cartoon. And also, we reach to almost the end saturation of the Moore's law at this moment, and there are physical limitation to slow down this increase of the computational power right now. So we probably, in my prediction, personal prediction, we will get into the quantum age. So Stone Age, Iron Age, and Silicon Age right now, but next probably is the quantum age. So this will be a question in your final examinations. 1, 2, 3, 4 ages, which one is not? What I consider, something like that. Okay, it's very easy. Don't worry about your exams. So this is a development timeline for quantum computing development proposed in 1980s, 90s. It's idea to use quantum computer to simulate quantum phenomena. And nowadays we have this gate-based quantum computer that you can probably to use as a general computational platform, but still people are try to figure out how to use this special features provided by quantum computer, especially this superposition and the entanglement properties. How can you use it wisely? That's a big problem. But still, people found some area to apply quantum computer, and not in biology yet. It's in the okay. So how to do it? We collect data and train the model, and evaluate the model. Once you finish, then you can apply to new data sets. Okay, this is very standard. So how can we learn this? So what we are supposed to do if you have zero background, use high school mathematics, and to learn this machine learning, we should start with linear algebra and understand some probability theory, and maybe a little bit of calculus. When you need to do the optimization, you know where to find your next point based on the slope, and algorithm and complexity, and the data processing. So some of those and combinations. Now, how can you learn? And a lot of this tutorial online, you can find, okay, we teach you the package to use. This is, I don't know, this is a TensorFlow, this is Python-based. And you will say, okay, it's very easy. You just import this and then do this, you are done. So this is not a good way to learn things, because you still don't know the principle behind it. I would say it's very useless, and you learn a lot of packages and how to write some of these pipelines, but you don't know what's going on here. Okay. And some will say, okay, it's very easy. You can do import SVM, then do the classification. Again, the same thing, and no matter how simple the code is, it's been heavily coded, and there are a lot of under the hood. So what I try to do also, okay, the PCA is very easy, okay, but make no sense. It's useless for us. Okay. On the other hand, this will be overloaded if you see. Okay, this is a gentle introduction of SVM, then they give you this. This is for not for biologists, not for us. So we have to be in between, and we will try to use simple language to explain the algorithm until you understand to the very basic, the bit. Okay, the computer is 0, 1, and you know how to do addition, and then you know how to do the multiplication. And then I will try to make you understand how from this 0, 1 flip until you do the PCA, rotate the data points until the very end, the application. So we are not going to do this simple PCA, not going to do this heavy mathematical formula-based. We will do using computer language at the same time to learn the principle, and you practice on the computer. And you probably can do this as a suggestion from a YouTuber, and there are different ways you can do, and I don't believe you should do those. And on this course, we use MATLAB, and okay, to do everything. You can use your own language if you still stick with R. Python is okay, but the principle will be the same. And we use MATLAB, but overall is like language agnostic. I don't care which language you use. So read some related research papers. We will have some examples during the class. Okay, so choose a language. By the way, MATLAB can call Python directly in MATLAB, so they try to make this communicatable very easily. I'll show you some. So we don't have time to do this. Maybe we can 5 minutes. Hold on.
Knowledge of machine learning algorithms. You should be familiar with popular machine learning algorithms
Okay, choose a language. I found some articles to compare MATLAB, Julia, and Python. So I believe Python is, which one is which? I don't know. This is F150? No, Julia is, so I don't know. You figured it out. Okay, and anyway, so we believe we are in the intersection of these two revolutions. One is machine learning and another is single cell data. So I will leave this for the next hour, and let's stop here, and I will come back to my MATLAB here, and my installation is finished. I don't know how about the yours, and you should be able to click this. You don't have to deactive, or it should be automatically activated already. And we will get into the interface of this large computational, engineering computational interface. It's just like a general application. You just click that button and they will bring up this window. I think today we can just have a very quick start, like get it started. And then you can bring back what you have today and apply it to your real application directly. I show you how. Without even know anything, okay? The first time user, you will get this yellow bar promote information, and then just getting started. If you click that, it will lead you to a very nice basic tutorial about the language, the environment. If you have never used the environment like this before, you should be able to get a good idea, okay? So now you have this command window. That's where you put your comment and then get feedback. And here is your memory, basically. Let me make font bigger. I make a bigger font to let you to see what I'm typing here. Yeah, any questions? Mm-hmm. Yeah, you shouldn't. Yeah. Okay, so it's in. If you click this, you should be able to start here. Yeah. Yeah, just wait. You should be able. Nothing happened. Yeah, it's just a wait. Yeah, some computers slower than others, and for some reason, yeah. Okay, you currently you are in the folder here. If you look at here, you are in C drive and user. This is my username, documents, and the MATLAB. So that's where your current working folder is. So this is the current working folder. This white space on your left upright here. There's nothing in this folder yet. And then here is your memory, and inside the memory, for example, you define a variable a equals 5, and you will see, okay, in my memory, I have a variable name called a, and the value is 5. Okay, so that's the way you look at it. Yeah, if you somehow lost, you can see. You can do this. If you lost, you can go layout here, and by default, it will set up back to this view. Okay, this is like your, by default, the view have these 3 panels. That's all. Okay, so in here you can input a matrix, which, for example, 1, comma 2, comma 3, and then semicolon 4, 5, 6, and then put back. So in this way, you define a matrix. This is a 2 by 3, okay? So I'm using integer, but if you have a decimal, and then it's okay. You have some precision, okay? So it's the same. So MATLAB, if you plus 3, and then every element inside the matrix will plus 3. So you don't have to go 1, 1. There's no loop, and it's just a matrix-based operation. Okay, today we just stop here. Okay, no language, but you can have a get started and to learn more by yourself. It's just assign a variable name and do some operation. So what I'm going to do is to directly just download the package we developed, and then start to do single cell data analysis. Okay. Yeah, so this toolbox is I developed from 2018, from 2019, so already several years. So this is named the single cell gene expression analysis toolbox. You can Google and click this documentation. It shows you the command line, how can you install it. If you Google and somehow found this page, that's correct. So you just copy this and paste. You highlight, copy this source code in this block. And paste into your command line. And then the program will just install this package and put in this folder. That's all you need to do. Then we are ready to go. So toolbox is a functional package MATLAB use to call the library. So it's toolbox, and this is a particular specifically designed toolbox for single cell gene expression analysis. Any questions? So here I finish my installation. So I just copy that and paste into common, press return, and this should give you a new folder in here. So that's the folder you install this toolbox. That's everything you need to do. That's all. Okay. So I will show you the interface that you can click, and then we will take a break and come back. Okay. So if you installed this, and then you can start to type lowercase. To bring up this example data set. Okay, so if you have this, then you are done. Then you can take a break. Okay? Okay, let's restart. So in this interface, this is allow you to get your data into the system, because you have to find a way to input your data in the system. You start to manipulate or working on that. There are different ways you can get that in. If you know you have your own 10x genomics output, I will show you how to get them there. You have a 10x genomics, the pipeline called Cell Ranger will give you either H5 file or the folder with serial files in there. So that's where you can input your own data set. If you have some data which other people generated in R or in Python, there's a way you can import them. Okay. There are also the way you can get directly online from published data. If this published data publish a paper, for example, in Nature Genetics, and there are many papers have a section called data availability, and where you can find geo accession number. Geo is a database in NCBI. Let me show you the geo. Where most of the NIH supported publication is compulsory to put their raw data and the process matrix data into geo database. So from here, you can, from the publication, to find this accession number, then you can search where the raw data is located. We can also, for example, if you really want to do a research on some of the cancer or epithelium cells, for example, you can get quite a lot of this results from the search. And then let me see, I put a 10x because I want to use the data generated from a 10x genomics as an example. Yeah, just randomly select a research, but this ID you should get from a published data, published paper, okay? Let me see whether this is the right data set. Okay, this data set looks fine for me. So I can have a quick look about this record in NCBI Geo database. First, you know this is from a mouse. Okay, remember we deal with human mouse data, most of the single cell data. And this study is about liver damage and caused by fibrosis, maybe. You can read some of the summary of the experiments to get some idea. And here you can see there are 4 different samples. And we are using sample level data. So you have to click inside here. For example, you know this is a injured mice liver cell isolated. So we need this ID. You can either copy this or you click go into and look at the details about this particular sample. So I just copied this, okay, right now. Copy, or you can remember to type in, and come back to this page. What we need is this number. This number is sample based. This is the number I just copied from that record. So if you provide this ID to the program, this program should go to the website to find that record and download the data set and prepare an process data for you. It will download that particular sample. You can also put the 2 samples together. You know, I have 4 samples. In this example, I have 4 samples. The first one is GSM 812216, another 1718. You can put multiple, okay, at the same time. So depends on the memory of your computer. If you put 20, you probably can overtake all the memory of your computer. So in this case, I will just, as an example, I will put these 2, okay? Remember the name is 1617. So in this dialog, I put these 2 IDs. The program will go to download these 2 and put cell together and start to compare, okay?
So we will select download and merge data.
It will, yeah, it will download and merge automatically. So in this case, I will click, I hope, okay, it will download and merge correctly. And demonstration is always hard. Some problem will happen, the network problem, but most of the case, it should work, okay? Okay, so I believe this is a network issue, so I have a solution. So I try single, and then if you have the same problem, we'll have to reset. Let's see. There are several. I know it's a connection issue. There's a Yeah, I will double check what's the problem with this particular data set. I randomly select an example, and for some reason, I just cannot automatically download this, maybe because of the naming problem, and it's very unstandard way to name it. Yeah, so let's just use the by default. There's another data set we can use just for demonstration purpose. So you can start to work on your own data if you have. So you click this and just use it by default. Okay, this should work. You can also, if you know you want to have 2 samples, you can get 8 side by side. Okay, this should allow you to download these 2 data sets and merge, and you have something to compare between. Let me know if you have any problem with this input, just by default, and then you copy that, and then put a comma between to separate them. Then you have a 47, 48. You can Google this ID, and you will find the original paper record. Yeah, it's a mouse islet, pancreatic tissue. If you're okay, you can click and just go ahead and let the program to do it. So you know the program will go to the website to find the correct files. It have to download. It will download an expression matrix plus the name of genes, and put the 2 matrices together. And then you will get the raw data which hasn't been processed. Then we'll show you how to do this 1-button click and to process it. Then this just gave you a quick way so you can start to work on existing data sets and to start to explore very quick and dirty ways to start a project. In front here, I have a slow machine, and you should have a better speed.
3-dimensional or 2-dimensional is 2D, right?
You can use 3D if you want to play with 3D. 2D is good for publication, but 3D give you one more dimension to look at the overlap of some cells. So for the exploratory, say yes, and you can do that. For demonstration purpose, I will skip this, and so I can get unprocessed data. Okay, otherwise it will be automatically processed. So that's the data I input from this study and the 2 records, And you see in total, we have 8,396 cells in this cell population, okay? I haven't rearranged cell by their similarity. That's because I haven't done anything yet, so it's just randomly put there. Those cells have been, each cell has been defined by 15,000 genes, okay? So you can click the first button here to look at the list of genes. Okay, those are the mouse gene. And you can use this label button to look at two populations. You click this and select batch ID. And the cells will be labeled with a color. One from one record, that's blue, another is from another record in red. They put together because, so you download the two data sets, two samples, and one blue cells from one and the red cell from another, okay? Now, the single button solution is within the experiment, then you can do single click solution, then you are done the analysis basically. So that's mouse, let's click. So it's
Is it this data that we cluster in? Like, does it look like this without being clustered separately?
that's right. I haven't clustered them, so they just randomly put them together.
So clustering would be a later step.
Yeah, so this is doing the clustering, everything. Yeah. So the question we can have right now is after clustering and rearrange cells, we will see whether the red cell and the blue cell from two different samples will be separable or they're still similar to each other, okay? So this is a good way if you know some papers and you want to double check their primary discovery, and you can run this yourself. Okay, yes, they identified 3 different major clusters, and here, yes, we identified 4, and they're a small one. There's some difference, and why you have a quick comparison. So this is a very easy way to, you haven't done any programming yet, but we will learn how to do more sophisticated analysis later. But for now, I just give you a very quick way, get your data in there, start to play with this, have some quick concept.
When I watched the data, they already clustered out, like, depending on the cell type.
How do you change the color?
The label.
Yeah. So this is unsupervised, based on the data itself to separate. We have a database to annotate the cell types automatically. They're doing it right now. Yeah, let's see what we can get from here. So that's my one-click button solution right now, and you can see this is from a primary tissue, and so you see multiple different cell types. You have a red blood cell that's supposed as a contamination because we don't care red blood cell most of the time, so we can get rid of them. But right now you have cells have been grouped based on their cell type similarity. That's from a pancreatic cell. So you have alpha beta cells, you have immune cells, okay, endothelium cells. There's a button here. You can click and start to rotate. Okay, this is a hidden button here. Okay. Yeah. The differences of this.
But how did you get it to show
Oh, how can I get this? Oh, so from unprocessed data, you can just, in the experimental, there's at the almost very bottom, there's one-click solution to let you to from raw data to this, okay? So again, so this small label allow you to label cells by either cell type or batch ID. So I can recolor them by their origin from which sample. Okay. Mostly they separated. Okay. Let's see. And then if you label them with cell type, you will have a different coloring system to allow you to see the color of the cells. Okay? So those are the very basic operation to allow you to have a quick idea about the separatable of your samples, what cell type are in there. Then we will consider more complicated analysis later. Okay? You can also use this big I view, and if you click this called multi-grouping, and you select batch ID and also cell type by holding your shift key and select these two, and then you can have this view and the side by side. If you rotate, they should rotate at the same time. And then you will see, okay, this particular cell type in tail color and have been separated into two clusters here by different colors. So give you this side-by-side view to understand the structure of your data. Okay? So I think today we will stop here, and all the buttons you can click because it only it crash your computer at the end. You can reboot. It's still okay. Okay. By the way, anytime if you see this small camera, you can click and make a video. And if, for example, in this view, I click this button here, and I will make a snapshot of video. It will make a MP4 file to save the view. Okay, inside this program, you need to know two data structure. One is this called a big X. This is a matrix. It's purely numeric. All the values of it's integer, so it's 0, 1, 2, 3. This matrix is gene by cell. It's in my memory of this, behind each of these. Another is a list of gene name. So you get big X and small g. That's all you need to remember. Small g. Okay? So I can show you these two variables in MATLAB here. You can click this button here to export the underlying data from this interface to your environment of the MATLAB. So you can see I will save the data into the workspace. Then you click X and G, and you will save these two variables. Once you do this and done this, then you will see the G is here. You have a list of genes. And X is sparse matrix, cannot display because it's too big. Okay? But I will force it to, then you can see this is a matrix. Okay, so you know under the interface you have two variables. G is a list of the string, which is gene name. And the big X is that spreadsheet like this matrix. All the values are in there. You see a lot of zeros, okay, because that gene's particular expression. So gene number 10's expression in first cell has not been detected, or this gene was not expressed in that cell, so that's 0. But gene number 11 expressed 2 copies has been detected, so it's 2. Yep.
It says that cannot display summaries of variables with more than 5
Yeah, because there's.
to open the X file.
Yeah, because so you can run this to make it work. You can force it to become full matrix. Remember to put a semicolon at the end, so you can see the matrix. You couldn't see because the data has been compressed into the sparse matrix format. But if you force the program to give me the full matrix, you'll still be able to see it. Because so many zeros, it was not memory efficient to display everything, but if you force it to display, it will let you know it's there. Okay, any other questions before we move to explain how this has been experimentally measured? So how can we get this from the single cell data sample? So I jump back to single cell technology. I will use 10x genomics droplet-based method as an example, but nowadays we have other options called parse bioscience. They use this multiplexing barcoding systems to separate cells. It's another different solution. So single-cell technology has been developed for many years. It wasn't such simple steps. So early stage, so this is 2009, and in called Spring Harbor, and this is a single-cell meeting. That's basically almost all the scientists working in a single-cell, just a few of them. It's a challenge, of course. Because we deal with small samples, there's so little mRNA in there. How can you amplify it? Over the years, we have a droplet-based and fluorescence, the separation, okay, and also microfluid chips. But at the end, we achieved from 2009 and this exponential increase of number of cells we can study in single study. And this data stopped at 2017, 2018, when 10x Genomics published their commercialized the platform. We start have a huge increase in 2017. So to me, the real starting year of the single cell biology is 2017, when 10x Genomics published their claiming this platform. They are still developing their product lines very successfully and penetrated into spatial transcriptome those days, and also flex kit that allow you to fix your cells and then wait until you collect enough samples and to barcoding them at the same time. But at the beginning, you have to prepare from fresh samples. So they make it more flexible right now. This is a single cell revolution, and science believes it started from 2018. This is a machine, it's called CRIM. And then you have a cell comes in, then make this oil droplet bubbles, and then capture the cell. So inside of this droplet, outside the oil, and inside the reagents, you allow you to capture this and the barcode, and you get the matrix from one sample. It wasn't possible, and if you use traditional RNA-seq analysis, you crash, smash your samples, millions of cells have been put together to get a vector of the data. That's one sample, maybe thousands of genes. Not very useful until you have enough samples, but you cannot reach enough cells like this level resolution. Never happened. So we are almost abandoned all the papers. I trashed all my papers published before 2017 talking about the gene expression, because at that time there's no single cell data available. You just put everything together, make no sense at all. I recycle all this printout. Another illustration, you have a droplet, and inside this is the size, okay? You see the small black dots in there. This the scale, the bubble is the droplet is in this scale in the cell. So inside the droplet, the secret is inside the droplet. You have a gel bead in the middle. I don't know how they make it, but each droplet in the center, this droplet, their gel bead covered with all the DNA barcode. Okay, each gel have a covered with the same barcode. For instance, if I have a gel bead in the middle, where's my? In the center, I covered with a lot of DNA oligos, and then but all the same sequence. If I have ACTTG, for example, I will have ACTTG all over. Okay, everything the same. So the cell inside will be later broken, and the mRNA will be released. And all the mRNA will be labeled with the same ACGDT, because it has been isolated inside the droplet, and everything in the gel bead covered with the same barcode. So everything released from this cell will be barcoded with ACGDT, this particular signature. That's why later, when we sequence them together, we will be able to recognize those mRNA from this particular cell, because they all labeled with the same barcode. We will be able computation to separate them. That's the trick. Is that clear? Yeah. Okay. So this barcode, so the right part is the gel bead. And this barcode is called a 10x barcode. Okay? So the example here, I only use 5 base pair, not long enough. In reality, it could be 18 or 21, I don't remember. But as long as you have enough base pair and the combinations of these different ACGT base pair, it will give you the enough resolution to separate the cells, millions of them. Okay? So that's a experiment procedures, and you barcode the cells and put in the drop oil, capture the cells and label it with the gel bead barcode and the lysis, and then PCR them at the end. So for this matrix, you can, for example, in MATLAB, you can build this kind of view, and this is a gene, the cell, the height indicates the level of this value. If it's 0, we just put level 0. If it's 1, in this case, is really high for this particular cell, for this particular gene, then you have 200, something like that. So they're all integers. They just count, okay? This is a view of selected corner of this particular population of cell, about 1,000, how many? 1,000 cells and 50 genes. And you see something like that. So this is a illustration I download from a company, and they try to use this bar plot to explain how we can use single cell data information. It's not very accurate because you don't know what's the height indicate for, but it could be a synthesized score, but each bar indicates a cell. And by looking at these cells, you know those from disease cells, those from healthy cells. And we want to understand this landscape by giving each cell some indicator markers and also some measurements. So that's information we can learn from an individual cell expression to define their states, right? The goal probably for us is try to find small molecule drugs or other perturbation treatment to push this whole landscape for the cell population, which in the disease states more towards healthy states. This is our goal. We try to learn this by looking at the population cells, whether any of these small molecule will have some impact for the population of cell. So if you have multiple cells from multiple treatment and put in this landscape, we probably have some hope to learn this procedure. So this is a very dynamic picture, but the illustration, just like that, we try to push cells gradually move to the healthy states. There are a lot of applications if you study, in this case, the zebrafish. If you have samples from a different development stage, okay, 4 hours after fertilization until 24 hours, then you collect multiple samples from different time points, and then you merge the cells and superimpose onto each other. You can see at the very beginning there are a lot of stem cell or embryo cells, and then later stage, you have a development of different organ, tissue, more developed cell types. This is from liver, so if you have a traditional RNA-seq from liver cells, because most of this tissue sample from liver cells, so you only get the information from liver cells, but in reality, you have all the endothelium cell, hepatocytes, blood cells in their immune cells. So you have a whole picture of the different cell types and their function, their interaction information you can learn from the single cell data. So that's the value of doing this. There are some challenges, as you said. You can see the matrix. If you don't do anything, there are 80% of zeros, or by count. Okay, zeros and zero one give you certainly information. A lot of zeros depends on where the zero is, and it will very informative actually. So we need to deal with this zero problems. This is called the sparsity, the stochastic variation. And visualize this high-dimensional data is also problematic because each cell is defined by thousands of genes. And if you have only 3 genes, it's 3D. If I have 4 genes, 4D, but you have thousands of genes, it's thousands of dimensions. To learn the relationship between cell types, define cell type itself is a hard problem. We have some hands-on experience. It's hard to differentiate the cell from this type to that type. For given markers, they are inconsistent across different definition. And how can you modeling? So modeling is a word that mean very different things to different people. If you have a computer background, you know the modeling, mathematical control for the data generation. It will be very different from your biology perspective, but we will learn and get a consensus later, okay? Modeling, how can you model the data? Basically, you want to know how can you randomly generate a data, which if you give you the randomly generated data compared to real data, you only tell the difference globally. That's a modeling. So you know how to model, to simulate, generate your observed data. Yeah.
I have a question about the matrix.
Yeah.
So when it has 1, 0, but it had 2 also in between, like different numbers were there.
Yeah, some housekeeping gene really high. If you look at the beta cell in pancreatic islet, the INS, this insulin gene, is shoot 2,000 times higher than average.
So
Yeah.
1 means that there is some expression and 0 means there is no expression.
0 means low expression, undetected, or maybe just shut down by the cell itself. So you have to differentiate by
So does it
some way.
take the previous literature, the data from previous literature that is available, and then gives it 1 and 0
Mhm.
regarding the gene?
Yeah.
What if that gene is not studied enough or known enough, like how the expression is? Then
Well, this method, at least by the original 3' kit, it's not a probe base. So you sequence it if it's there. It's not you design a probe, will detect or not. So it's not based on the pre-knowledge of the probe. But I don't know the Flex kit. The Flex kit is based on the pre-designed. So they probably have a different coverage of the genes. Yeah. So that's why people start to use different ways to do this dimensionality reduction. PCA, very important fundamental method, but people found for this complex data set, use t-SNE or other method, newly developed data scientist method will be better to separate the cells. We nowadays can get the expression level and also attack seq, which is epigenetics. This is called chromatin accessibility data as well. So if you more ambitious, you can have single cell ataxic side by side with your cells. So those are the open chromatin structure. So you have another big matrix Y. And how can we learn from this interplay between two matrices for same sample will be very challenging. And people start to working on this so-called multimodal, okay? Your sequence from same population of cells, same cell, you have a single cell RNA-seq, that's gene expression level, and also single cell ataxic data, and the open chromatin structure information from that cell. And what we can do with this, another challenge problem. Okay, so this is the idea about pseudo time. We can cluster cells into population. We can also arrange cells by their similarity. So you put one cell here, and you find another very similar to it to the next, and then you find another highly similar to you. Make a linear arrangement of the cells, then you can see, okay, from this end of the line to that end, there's some development stage can be recovered by the rearrangement of those cells. You can learn from there, say whether there's gene start to upregulated across the development time, or some is downregulated, some maybe shoot high in the middle. So that's the way people will think about how to use the single cell pseudo time analysis. You align cells and assign a development time, which is not real, but by the similarity. You should have a small steps to move from one cell to another. Okay, those are the places you can find the data. So we mostly found from the published data from a geodatabase.